Thank you again for the invitation to speak today. I'm excited to talk some about my journey and work in pharmacogenomics. And so I was born and raised here in Alabama. I grew up in Alabaster, which is about 30 minutes south of here. So as you can kind of see on the slide, my academic journey is very concentrated in Birmingham. I got my undergraduate degree in biology here at UAB. And then for the next 4 years, I went on to work in a cellular molecular pathology lab, really focused on breast and prostate cancer. And so this gave me really a strong foundation in research. I learned about genetics as well as exposure to gene therapy-based treatment options. So I really wanted to further my understanding of pharmacology and have a more direct impact on patients. So that's when I went back and got my PharmD from Stanford. And so during pharmacy school, my interest in pharmacogenomics really took off. Towards the end, my grandmother was diagnosed with lung cancer, and I found myself really diving into the literature to learn anything I could about targeted therapies that target her specific mutation. And so that experience really opened my eyes to the power of genomics to guide drug therapy. But at that point, I didn't really know how to pursue pharmacogenomics as a career. We learned a little bit about it in school, but that was kind of it. So I Googled UAB and pharmacogenomics, which led me to Dr. Nita Lindy, who many of you may know. And so she's also a pharmacist and a chronic disease epidemiologist. And so she's made major contributions to identifying genetic variants that affect drug response and then translating those into clinical gene drug guidelines. So after meeting with her, I pursued the T32 fellowship in genomic medicine under the mentorship of Dr. Lindy, and she has been my mentor ever since. And if you take one thing from this talk today, I would recommend finding a great mentor because it has really made all the difference in my journey. So after my fellowship, I then transitioned to an instructor, and then in 2023 to an assistant professor. And so broadly, my work focuses on translational pharmacogenomics. And so that's from identifying genetic predictors of drug response to the clinical implementation of associations that have sufficient evidence, and then evaluating the population level impact. And so I am going to talk briefly about each of those areas today, but first I want to introduce variable medication response and give you some key kind of pharmacology and pharmacogenomics concepts to kind of frame the discussion. Then I will go into our research on genetic predictors of CVD response in epilepsy and close by showing how we translate pharmacogenomic discoveries into clinical practice. And I know that is a lot, so if at any point anyone has any questions, just feel free to ask. So our medications really form the cornerstone of therapy for the treatment of chronic disease. But our current approach to therapy is largely one size fits all. So we take patients with the same diagnosis and give them the same medication at the same dose and hope that they have a beneficial response. But in reality, response is highly variable. So some patients see little to no benefit, while others can experience exaggerated side effects or toxicity. So unfortunately, variable response to medications is very common across drug classes. Anywhere from 38 to 75% of patients don't respond to the first medication they're given. And then on the other side of this, adverse drug events are now the third leading cause of death in the US. And this was actually just recently upgraded earlier this year from the fourth leading cause. And so annually, adverse drug events account for 1 million emergency visits, 2 million hospitalizations, 3.5 million doctor's office visits, and $136 billion in US healthcare costs. And importantly, these aren't patients who were given the wrong medication. These are patients who, by current guidelines, were treated appropriately. And so this really highlights the real world impact of what variable response can do. So ultimately, our goal is to deliver the right medication at the right dose for the right patient. And we know that factors like age, weight, sex, and kidney or liver function influence drug response. But even when we account for these, variability remains high. And so it's suggested that 20 to 70% of this variability may be explained by genetic factors. And this can depend on the specific drug or outcome that's being evaluated. So pharmacogenomics combines genomics and pharmacology to understand how our genes influence medication response. It can help guide dosing, prevent adverse reactions, improve efficacy, and even support new drug discovery and development. And so by incorporating pharmacogenomics into clinical care, we have the real opportunity to personalize treatment, reduce the trial and error approach to prescribing, and improve medication and health-related outcomes. And so before I get into my research, as I mentioned, I just want to discuss some core pharmacology principles and how these relate to genetic variability to help frame the discussion. So there are two major components to pharmacology. This is pharmacokinetics and pharmacodynamics. So pharmacokinetics, or PK, is essentially what the body does to the drug. So this is absorption, distribution, metabolism, and elimination. So after you take a pill, it's absorbed through the gut, distributed to the tissues, and then metabolized primarily in your liver, and then it's cleared through the urine, bile, or other routes. And so pharmacodynamics, or PD, is what the drug does to the body once it reaches its target. And so this also includes how we distinguish between therapeutic and toxic effects. But genetic differences can influence both of these PK and PD pathways and affect how drugs are processed in the body and how they act on their targets. And so this can lead to higher or lower than expected drug concentrations and differences in efficacy and or safety. And so generally we understand pharmacokinetics better because we can measure drug levels. But it's difficult to study drug target effects, especially for medications where we don't know how they work. So pharmacogenomic variability is very common. Nearly everyone has at least one clinically actionable variant. And so the figure on the left really shows population differences in variant frequencies for 5 major drug metabolizing enzymes. So these genes are really responsible for the metabolism of about 70 to 80% of drugs currently on the market. And so you can see on the right how these population frequency differences translate to differences in metabolism. And so you can have poor metabolism shown in orange, and then that ranges to ultra rapid shown in blue. So on the other side, genetic variation also affects the drug targets themselves. So this study looked at genetic variation in over 600 drug target pairs. And this is only about 19% of total known targets. And they found that about 1 in 6 people carried a variant in a drug binding site. These carrier frequencies also differed by ancestry. And so taken together with what we saw from the previous slide, it really highlights that genetic variation is common in both our PK and PD pathways, and these can meaningfully influence how we respond to medications. So biologic sex can also influence how patients process and respond to drugs. And the figure on the left shows differences in drug pharmacokinetics by sex, and each example actually shows a different drug. And so these differences are clinically significant and common, and they've been associated with higher occurrence of adverse drug reactions among women. So sex-related differences in gene expression may explain some of this variability. So over 700 genes in the liver, which, as I said, is where most of our drug metabolism occurs, shows sex-biased expression, and that includes 77 genes specifically involved in drug response. And so on the right, you can see that a number of these pathways are specifically highly enriched in females. So all of that variability really sets the stage for pharmacogenomic association studies. So where we try to link genetic variants to differences in drug response in patients. And so by comparing patients who experience a therapeutic effect, an adverse event, or a biomarker change to those who didn't, we can identify a specific variants that contribute to altered response. And so these studies are typically done using either a candidate gene or a genome-wide approach, or GWAS. And so currently many pharmacogenomic studies have used candidate gene designs. So they select a handful of genes that are thought to be important based on the drug label or known biology or prior studies. However, this approach can really introduce bias in gene selection, and we can miss unknown pathways or unknown targets. And it has limited potential for identifying novel associations that could clinically impact response. So on the other side, you might be familiar with this, but in a GWAS, we scan the genome to link variants with drug response. But the issue with this is these need large sample sizes, and in pharmacogenomics, patient numbers are often limited, and defining drug response precisely can be tricky. So drugs in the same class might not go down the same pathways, like I showed you earlier. And so if we group them, it can dilute important genetic signals. But on the other side, if we analyze a single drug, then we reduce the sample size. So when you look at sample sizes in GWAS, pharmacogenomics GWAS have had a median of about 1,200 participants. And this has stayed relatively flat over time. While on the other side, GWAS have grown steadily, peaking above 10,000 participants. And this is showing pharmacogenomics GWAS in comparison to any other GWAS. And so interestingly, in pharmacogenomics, larger sample sizes don't always mean more discoveries. There's actually only a modest correlation between an increase in sample size and the number of significant associations. And that's shown on the bottom right. So what's missing here? It seems like it may not just be about increased sample size. Well, we also need better phenotyping, mechanistic insight, and then increased diversity to really begin to understand how this actually impacts drug response. So this really led me to an important question, and that has guided much of my research into identifying genetic predictors of drug response. And that is, can a comprehensive drug-related gene approach help us understand variability in response, especially when the mechanisms are unclear and candidate gene studies may miss important biology? And so one of the first pharmacogenomic association studies I led explored genetics of response to cannabidiol, or CBD, in patients with treatment-resistant epilepsy. So epilepsy is a neurologic condition characterized by recurrent unprovoked seizures. It affects about 3.4 million people in the US, and about 1 in 26 people will be diagnosed with epilepsy at some point in their life. And so on the right, you see available anti-seizure drugs. And if you look over really the past 30 years, about 25 new drugs have been introduced. However, epilepsy outcomes at initial diagnosis have not really improved despite all of these therapies. So on the figure on the left, this shows the probability of seizure freedom by the number of drug regimens tried. So about 50% of patients achieve seizure freedom with their first drug, but the probability of seizure control decreases substantially with each additional regimen. So after 2 failed drug trials, the probability of control is very low, and this group is classified as having treatment-resistant epilepsy, or TRE. So TRE affects about 1/3 of individuals with epilepsy, and it's associated with increased healthcare utilization and worse outcomes. Therapies that can improve seizure control for these patients are desperately needed. So Epidiolex is a plant-derived, highly purified form of CBD that was FDA approved in 2018 for 3 rare epilepsy syndromes. The CBD Expanded Access Program, or EAP, was a compassionate use open label study that was conducted at 35 US epilepsy centers from 2014 to 2019. And the goal of this was to provide CBD to patients with other highly resistant, treatment-resistant epilepsies. And so UAB was one of the participating centers in the EAP. And to enroll in the UAB EAP, patients had to be an Alabama resident and meet really strict criteria. So specifically, they had to have confirmed TRE based on 3 different metrics, which were video EEG monitoring. They had to have at least 4 seizures per month over a 3-month period. And they had to have tried and failed at least 4 different medications, including a trial of 2 medications together. And so this criteria was much more stringent than the overall EAP population. And so this really provided a very well phenotyped group to conduct additional analyses. And so overall, there were 169 patients in the UAB EAP, and about 64% achieved at least a 50% reduction in seizures. And so this was really, beneficial. These patients had to have tried and failed at least 4 therapies already. So this was a significant increase in response. So we really wanted to know, are there any clinical predictors that can predict who responds to CBD? But aside from CBD dose, there were really no other clinical factors that predicted response. So we wanted to understand if there were any genetic predictors that could affect response. And so EAP patients were approached for enrollment in the genomic study, and a total of 112 participated. Patients underwent whole genome sequencing, and then they also were genotyped on a pharmacogenomics focused array that included about 230 drug-related genes. And so if you see on the right, the majority of these genes are involved in the pharmacokinetic pathways. So essentially, this is going to give us some insight into genetic factors that influence metabolism and transport. This array really doesn't include any of the drug targets. So CBD response, as I kind of alluded to, was defined as at least a 50% reduction in seizures, and this was based on the trial definition. So in the genomic cohort, there were 56% of the patients were classified as responders. So we looked at baseline characteristics. They were similar between patients who responded and didn't respond. The main differences were in, you can see here outlined in red, were in CBD dose, and then side effects like diarrhea and abnormal liver function, which was seen more often in responders, and then sedation, which was seen more often in non-responders. And so on average, patients in our genomic cohort were in their early 20s. They'd been diagnosed with epilepsy around the age of 5 or 6. And what's also interesting here is they had tried about 8 prior therapies, and then we're taking 2 to 3 other drugs along with CBD. So as I said, our trial was much more stringent. So these patients had tried an average of 8 therapies prior to CBD. So this was initially planned as one of the candidate gene studies. We were going to review the literature and see if there were a handful of genes we could select to look at. However, CBD metabolism is complex. It is not fully understood. There are multiple enzymes that can metabolize CBD, and over 40 metabolites have been identified in humans. And the other thing is that the mechanism of action is unknown. We don't know how it works. So couldn't really select. So I wasn't comfortable selecting any of these candidate genes. So we sort of adapted at that point and made this use the comprehensive pharmacogenomics approach that I referred to. So we included all of the genes on the array. But again, most potential targets weren't included. And so we used permutation testing to account for multiple comparisons, since these genes were all in drug-related pathways and likely not independent. And since this was really intended to be a hypothesis-generating study, we felt this was the best approach. So I'm not going to go into the overall results because we did report all significant associations. But for genes and variants associated with response, we wanted to see how these might affect gene expression and drug targets to understand any potential underlying mechanisms. So these patients are highly treatment resistant. Is there anything that we can find that is leading to why they're resistant to all of these therapies? So at the gene level, we asked whether these genes are co-expressed and if this could suggest potential pathways underlying multi-drug resistance. And then at the variant level, we evaluated whether any variants influenced expression of potential CBD targets in the brain. So there were 29 genes associated with CBD response. And when we looked at this co-expression network, 59% of them were co-expressed. And these are shown in the figure here outlined in dark black. And so what was really interesting was that 75% of the genes that were associated with lower odds of response were co-expressed, compared to only 38% of those that were associated with a higher likelihood of response. And then notably, some of the pathways that emerged in this network had roles in neuronal processes. For example, a Notch2 NLB is actually a human-specific gene that plays a critical role in brain development. And it was found in this network when we included only these drug-related genes that were associated with response. And so really, even though we only looked at these pharmacokinetic genes, when we evaluated co-expression, it allowed us to identify potential pathways that could be underlying therapeutic resistance. And so next, at the variant level, we looked, if any of these variants associated with response influence the expression of potential CBD targets in the brain using the Brainiac database, which was from the UK Brain Consortium. And so we found several interesting effects, but two really stood out. And so the first shown on the left, that variant actually decreases, and both of these are associated with lower odds of response. And so the first variant shown on the left decreases the expression of GPR18. So this is an endocannabinoid receptor, and it's actually structurally similar to one of the proposed CBD targets. And so had we selected candidate genes, if they had been available, our targets, we probably would have selected that other endocannabinoid receptor, not this one. And so the other shown on the right, this variant actually decreases the expression of HTR3E. And so this encodes the serotonin 3E receptor. And this receptor might influence neurotransmission, and it's also highly expressed in the gut. And so could this be why we see diarrhea more frequently in responders? And the other big thing to note here is that HTR3E is absent from most rodent genomes. And so this effect would not have been observed in animal studies, which is also similar to the Notch2 NLB. It's also a human-specific gene. And so our sample size was very small. These findings really highlight potentially novel human-specific biology and that may be missed in traditional models. So building on the concept that drug-related genes can reveal underlying biology beyond just our drug metabolism and transport, we asked whether a similar comprehensive pharmacogenomic approach could be applied to other high-impact medications. So anticoagulants or blood thinners are one of the top most prescribed drug classes in the US, but they're also the top drug class implicated in patient harm. And so you can see these top drug classes on the left. And these are responsible for emergency visits related to medications. And so when we look at variability in anticoagulant response, this can really have detrimental consequences. If you have too little, then you can have thrombosis. But if you have over anticoagulation, then you can have hemorrhage. And so as Dr. Urban mentioned, I currently have a K-23 that is focusing on anticoagulant pharmacogenomics. And so in this project, we're using a similar approach, really guided by what we did with the CBD work. And we're also integrating this functional information in addition to metabolomics to see if we can identify predictors of pharmacokinetic, pharmacodynamic, and clinical response. And specifically, we're also paying attention to the things I mentioned, like ancestry, sex, and age-specific effects that are known to affect how we metabolize and transport medications. And so, although this work is ongoing, so I don't want to focus on it too much today, the ultimate goal is to bring precision medicine to anticoagulant prescribing and make this high-risk therapy safer for everyone. So these genetic association studies really give us insight into mechanism and potential biomarkers that affect how we respond to medications, but discovery alone really isn't enough. So we have to take this knowledge and apply it in clinical practice to improve medication dosing, selection, and outcomes. And so this is where the clinical implementation piece comes in. So translating these findings into tools and guidelines that clinicians can use to make more informed, safer decisions. So research has identified thousands of variants that affect drug response, but not all of these have the same impact. And so the figure on the left shows the number of newly discovered pharmacogenomic associations each year by drug class. And so the field is growing, but this also provides us with a lot of information. So how do we know what's clinically relevant and what needs further research? And so ClinPGX is a comprehensive pharmacogenomics resource that helps organize and make sense of all of this information. It catalogs, curates, annotates, and disseminates pharmacogenomic knowledge. And then it gives clinicians, labs, researchers, and patients a centralized place to access pharmacogenomic information. So when deciding which gene drug pairs to implement and return clinically, we really rely on two key resources. So this is CPIC, the Clinical Pharmacogenetics Implementation Consortium. And so this group develops peer-reviewed, freely available gene drug guidelines to guide clinical use. And then the FDA, which provides pharmacogenomics information in drug labeling updates. And then they also maintain a table of pharmacogenomic associations, which are things they consider to have sufficient evidence to guide clinical therapy. And so we also consider whether testing is actionable. Is this result going to lead to a prescriber changing therapy? And then there are other factors. For example, is this testing covered by insurance? And insurance actually does cover some of this testing for very specific criteria. But these help us prioritize what we should implement in clinical care. So this figure just shows the total number of actionable gene drug associations recognized by both CPIC and the FDA and the overlap between the two. And so these resources really complement each other and help guide which associations we can use in clinical practice. So here are just some of those, because I know that was a large list. Here are some of the clinically actionable medications really across therapeutic areas. And the ones shown in red are drugs that are among the top 300 most prescribed drugs in the US. So these are really commonly used medications across therapeutic disciplines. So pharmacogenomics really has broad clinical relevance. So because so many of these medications with clinically relevant gene-drug interactions are commonly prescribed, we wanted to understand how impactful pharmacogenomics could be at the state level. So the Alabama Genomic Health Initiative, or AGHI, is a state-funded initiative that was launched in 2017 to harness the power of genomics for disease prevention and treatment. And so to date, over 10,000 participants have been enrolled from all of Alabama's 67 counties. So we leveraged genetic and medication data from roughly 4,200 AGHI participants who had a UAB clinical encounter to evaluate pharmacogenomic actionability really across the state population. And so here you can see the breakdown of actionable genotypes and medications. And not surprisingly, you see the CYP metabolizing genes we discussed earlier. But the key finding here was really in line with other national data that almost everyone has at least one clinically actionable genetic variant that could be used to inform future prescribing. And so this allowed us really to quantify the prevalence of actionable variants and how this information could be used to influence prescribing decisions in the real world. And so these results were compelling enough that in 2021, AGHI actually added pharmacogenomic testing and return of results. And so we now currently do pharmacogenomic testing and provide patients and their providers with these results. And so AGHI really illustrates the utility of preemptive testing. And so this is having results available before a prescribing decision is made. But clinically, pharmacogenomic testing is usually done reactively, and this is largely due to insurance coverage that I mentioned earlier. So reactive testing is usually ordered after a drug has already been prescribed, often in response to an adverse event or failed therapy. And so our clinical service here is primarily reactive, but through AGHI, we've been able to preemptively genotype over 10,000 allobamians and integrate the results into the medical record. And so later, if one of these patients is prescribed a drug with a gene-drug interaction, that information is available to their provider, and so they can make an informed, safer decision. So this really bridges the gap between research and clinical care. So the last thing I really want to walk through is how we built the infrastructure to make clinical pharmacogenomics actionable in practice here at UAB. And so as we worked and moved towards clinical implementation, our first priority was making sure that our genotyping panel captured all clinically relevant genes and alleles and was representative of our diverse patient population. And so the commercial array that we started with that's shown here in the middle included a standard set of pharmacogenes and variants. But we recognized that there were some important genes and variants missing, particularly some that are more common in non-European populations. And so without these, we risked misclassifying phenotypes and potentially recommending inappropriate therapies. And so to address this, we evaluated all of this, compared population allele frequencies, identified these missing genes and variants, and designed a custom array. So now our core panel and custom array are used for genotyping here in our clinical lab. And so the panel you see on the right reflects the genes and variants that we added. So initially, these pharmacogenomic results were uploaded into the EHR as a PDF. And so while this captured the data, the clinicians often overlooked them. They were sort of lost in a sea of documents. And they averaged around 30 pages long. And so providers said, I have no idea, what to make of this. And so this was sort of a multi-step process, but our first step was really to create a dedicated pharmacogenetics reports folder in the EHR so we could centralize these reports so they could at least be located. And so we did that. We worked with our IT and informatics teams to build that. And so this was an improvement, but the results are still PDF and not really structured for clinical use. So our goal was to capture these results as discrete structured data. So we went back and worked closely with IT and informatics to build this PGX flow sheet, which is shown here on the left. And so this allows us to enter the genotype and phenotype information in a way that's really user-friendly, and it can also be machine readable. And so on the right, this is how this information now appears in the patient's chart. So you can quickly see their metabolizer status and or the genotype if you want to. And so this allows this information to be easily viewed And it also allows it to be linked to clinical decision support, so clinicians can be alerted to any actionable gene-drug interaction at the point of prescribing. So for the clinical decision support piece, we integrated ActX, which was a clinical decision support software, into our medical record. And so this reads the genotype data and provides a dynamic interface shown here on the slide, where the clinicians can review any of these gene-drug interactions. And so this can also be tied to real-time alerts, providing actionable recommendations like dose adjustment or a different medication. And so our team customizes all of the content within ActX to ensure that providers receive evidence-based guidance without being overwhelmed with unnecessary alerts. And so as part of this, we also measure uptake of pharmacogenomics recommendations, any prescribing changes, and then patient outcomes. And then we also work with our clinicians to gather feedback and refine the system to ensure that it's useful to them. Because we can design it how we think it'll be useful, but getting their feedback and really refining it is that extra step. So this is just a high-level overview of our clinical pharmacogenomics service. The test is ordered in the EHR, the sample sent to our lab for genotyping on our custom panel, and then the results are returned both as discrete data and through ActX. And so our PGX team then reviews the results and provides a consult with actionable recommendations that also considers these genetic results in the context of other patient-specific factors like their allergies, current therapies, organ function. And so this is our clinical pharmacogenomics team. We work really across both research and clinical care. So we advance pharmacogenomics and really genomics and general research while integrating actionable findings into clinical workflows so that pharmacogenomics can have a real-world impact. So I hope I have shown you today how pharmacogenomics really spans the translational research spectrum, really from discovery to clinical implementation and refinement and reimplementation. And so pharmacogenomics really has the power to improve health and medication-related outcomes, from helping identify why one patient doesn't respond to therapy or another patient has a severe adverse effect. And so by understanding this genetic variation, we can move closer to delivering the right drug to the right patient at the right dose every time.